Right, so we're just picking up here where we have been talking about parsimony, right, and how to create a tree. And so you should all know how to do that. And remember, there was this problem that there are lots of trees that could be possible, right? And the parsimony tree is the one that requires the fewest numbers of steps. So to find that tree, remember, we just count up all the possible ways in which you can map characters on all of the trees. Find the tree that has the shortest length, the one that has the fewest number of changes, right, on the tree. And remember that we said that there's this huge number of trees, especially as your number of taxa get bigger. You know, where there, by the time you get to 50 or 60 or 70 taxa, there's more possible trees than there are atoms in the universe, right? So it's this huge problem. So we talked about how you can do branch and bound searches, and that was part of the assignment. And then we also did taught you how to do heuristic searches, which is what, if you're going to do parsimony searches for your projects, you're going to have to do heuristic searches. So remember, we did this analogy of a plane flying over a landscape of possible trees and trying to figure out which one of those trees is the best tree, with the parachuters coming down and they're walking around and they're searching to try to get to that top peak that has the best tree. Now, sometimes that results in lots of most parsimonious trees, so trees that are all the same length. So then you've got to figure out the majority rule consensus of those trees. And so, if we looked at the top one here, we would say, is A and B found in these other trees? And it is found in the third tree, but not in this tree. So therefore, it's found in 67% of the trees. The node that contains A, B, C is found in all three trees, and so therefore it has a consensus value of 100% and so forth. Or you could do a strict consensus where these are essentially just 100% consensus values. And I'll show in a video on the side how to compute consensus trees. So we've talked about the advantages and disadvantages of parsimony. I'm not going to go through that. The next set of methods is distance-based methods. And I'm going to quickly go through these because we already did something very similar to this before, where you do a comparison side by side of two sequences. Here we're looking at amino acid sequences. So the differences between human and cow, we see that the differences are shown here wherever there's a letter. If there's a dot, it's the exact same as the one above. And so there are 1, 2, 3, 4, 5, 6, 7 differences between human and cow. The number of common sites, there's 10 nucleotides, so there's 7 of those blocks. So there's 70 total nucleotides, 7 of them are different, so that means the p distance is 7 over 70 or 0.1. And again, remember, we can look at more complicated ways of trying to get the real evolutionary distance. You can start off with the Poisson distance in amino acid data, then you could use PAM and PAM matrices or blossom matrices to do this. If we were doing DNA, then you would do models. You would do things like the D distance, and then the Jukes Cantor, and then the Tamura parameter 3 model, and then eventually the GTR model, right? But here we're just looking at amino acids, and we're going to just keep it nice and simple. The comparison of human and kangaroo, if you count up these number of differences, there's 15. So the p distance would be 0.214, and the d distance would be 0.24, right? A more correct evolutionary distance. And then you could do the comparison of cow and kangaroo and come up with those distances as well. So you can summarize all of that in a matrix. We know that the matrix is symmetrical, so the numbers on the top are the same as the bottom, and the ones along the diagonal are just always going to be zeros. So you can get rid of all of those, and you end up with your pairwise distance matrix. Then you simply just say, all right, how similar or different are these things? And for simplicity's sake, let's just talk about number of differences. So the difference between human and cow is 7, human and kangaroo is 15, and cow and kangaroo is 15. So if I look at that, we assume that evolution is occurring at a relatively constant rate, and that differences are going to accumulate at a constant rate. And if this is true, then what does this say about the time since they've diverged? Well, it means that the same rate of evolution has been happening since time of divergence. So I know that the human and cow, therefore, have the most recent divergence, right? So I can draw that with branches and a node. And you can even put the branch lengths on there. If you assume that it's the same rate on each side, it has to be 3.5 differences per side, even though you don't really have 3.5 differences, but that's okay. This is an evolutionary distance, and you can divide it up that way. And that now becomes that lineage that we would call man-cow, right? Now, once we've done that, then we can simply take the average distance of human to kangaroo and cow to kangaroo, and the average of 15 and 15 is 15. And so we have a new man-cow to kangaroo distance of 15, and then we put that on our tree, and then the distances on the side. And then you can essentially make the full tree by putting the two pieces together. So that tree, these branches and nodes come together, and they combine with this one, and you essentially get now the full tree of human, cow, and kangaroo. This is called the unweighted pair group method of arithmetic mean, or UPGMA. And this is one of the methods you can use in Mega. So to do this, then, again, let's just talk about the steps again. You find the pair with the smallest difference. You join them in a group. You compute the average distance between this group and all other taxa one by one. You repeat those steps using the new distance matrix, and you just keep going. Okay, so here's a more complicated example of this. So up here, which two pairings have the least amount of differences? B and D, right? They only have two. So I put D and B together, okay? And there would be the differences 1 and 1 on each side. Then I have to do the average difference of D to C and of D to A and B to A. So D to A is this one right here. D to A is 3, and B to A is 5. So what's the average of 5 and 3? It's going to go right here in this cell. It's 4, right? And then D to C and B to C, and its average is 9, and D to E and B to E, and its average is 7. Then you ask the question again, which two pairs have the shortest distance? And it's 4, and so therefore I'm going to put the A to the D and B, okay? And then put my branch lengths on there as well. And you just keep doing this process. So now I do ADB, average to C and E, get those new averages. The smallest is 6. So now I put C and E off to the side. And that's okay, it's fine. Eventually they're all going to come together. And keep working my way down through here. And the average between C and E to the ADB is 9 and 7, which is 8. And so then you get your full tree together, you finish all your distances, and that's how you make a distance tree. Okay. Now, obviously, the assumptions here is that this requires that the tree is additive, that all distances between any two taxa are equals the sum of the lengths of the branches is between them. It also assumes ultrametricity, that all taxa are equally distanced from the root. And these assumptions, of course, are going to be violated, and so in a lot of normal data. And so in distance methods, there's a method called neighbor joining, which tries to minimize some of those assumptions and allows for branch lengths to be variable and allows for the trees don't have to be ultrametric and so forth. So this is a much more popular method, okay? And it uses this doing a slightly different version of the logic that we just went through, but it's pretty similar. And so it does these steps where you take each taxa, you calculate the net divergence from all other taxa, you use the net divergence to construct a corrected distance matrix, you find pairs with the lowest corrected distances, you put those together first, you get a new matrix, you keep doing that. So it's similar in the logic that we just did. It just allows for corrected distances on each branch, where branches can be different lengths, and so you can have different rates of evolution happening on each branches. Okay. And I'll show how that's done also in a different video on how you do that in Mega. And then the last methods are maximum likelihood and Bayesian. Now, maximum likelihood and Bayesian, essentially it's where you're trying to find the tree that maximizes the probability of observing the data on that tree. To do this, though, you have to have an evolutionary model, okay, that gives the rates that each of the 4 nucleotides are going to change into. And we've already talked about models, okay? So you can use the model choice program to do that. As with parsimony, though, we've got to check all possible topologies. So you still have to calculate every single possible topology for the likelihood across all of those different topologies, and then say which one is the best. So to do this, if we just look at a very simple case here, where you have the 4 taxon unrooted topologies, so there's 3 possible trees here, you have to ask the question, what is the probability that a particular tree would have generated the data presented in the sequences under that chosen evolutionary model? Okay, so to do this, you calculate the likelihood for any site, and we have to consider all possible ways that nucleotides at the tips could have evolved. So that means that you have to consider that if there's a T present at the tip, it could have been, though, an A that once was a G, and then, or that went to a G and then went to a T, or it went from an A to a C to a T, or an A to a or an A straight to a T. You have to look at all of those different possibilities. And so for every single node, you end up looking at lots of different possibilities. And so you have to then add up independently all of the likelihoods for every single site, for every single node, for every single tree. And so you get these huge calculations of likelihood that are taking place. And at the end, it becomes a really, really small number, because the likelihood of each one of those individual circumstances is a really small likelihood. So you get this small likelihood score. Since these are really small, we do the log likelihoods at each site and report the likelihood of the entire tree as a log likelihood. So this ends up being a negative number. So if we compare all of these methods together, neighbor joining is really fast and can work on really large data sets, but it tends to make mistakes and tends to not do as well at reconstructing the correct tree. Parsimony is reasonably fast. It does okay with large data sets, but it sometimes gets it wrong. In fact, it gets it very wrong if branch lengths are if there's different evolutionary rates across different branches. Parsimony does not perform well there. But it's the only method that can really work with non-molecular data in any real sense. And so it's still a very popular method, especially if you're going to be doing analyses that combine morphological and molecular data. Maximum likelihood is really slow, but it's actually the most accurate, and Bayesian is thought to be kind of a faster version of likelihood. Now, I won't be teaching Bayesian this semester. We don't have time to do that, but I will be teaching a video that shows how to do likelihood as well. So by the end of this, you should have already known how to do parsimony. I'm going to show you how to run through some of these other methods in Mega as well, so that you're comfortable doing all of these methods. And the last thing I want to talk about, and I'll do also a video that'll talk specifically about bootstrapping, is bootstrapping. But this is a common method of estimating the confidence in phylogenetic studies. Okay, so it's a statistical technique in which one randomly samples from a data set with replacement the different sites. So imagine if you have a data set, with 10 sites. You need to randomly sample those 10 sites, but with replacement. So what's going to happen is you're going to get a new data set of 10 sites, but sometimes you're going to represent one site more than once, or one character more than once, and some sites or some characters will not be sampled. Well, if you do that, you're going to end up then with these data sets that over-represent characters and under-represent characters. And then you reconstruct a tree for each one of those resampled data sets. And maybe you resample 100 times. And then you just calculate how many of the 100 data sets ended up supporting a particular relationship. And that's the node that goes. So it's essentially, remember we talked not too long ago about consensus trees. A bootstrap analysis is a consensus tree of 100 or however many times you do the bootstrap. It's a consensus tree of all of those replicates of the relationships that are found in those 100 replicates. The bootstrap value is a consensus of those. Okay, so you do this. If the original data set has an n characters, you sample those n characters randomly from your data with replacement. Some characters are going to be chosen more than once, and some are going to be skipped. You estimate a tree based on that on the sample data set. You repeat that for the number of times that you're going to do your bootstrap analysis. I think the minimum in Mega is 50. And then you calculate the majority rule consensus from all of those replicates. The majority rule value on each branch becomes the bootstrap support value. And that indicates how well the data support each of those specific branches, each of those specific nodes or clades. All right, so look for the individual videos on how to do each one of these in Mega. And that's the end of the lectures that we'll be doing for the semester.